Okay, thanks everyone for joining us today. It looks like there are many people still joining, but let's get started anyways. My name is Maija Itkonen. I'm the CEO and co-founder of Onego Bio. For Onego, this is a very special day. So it's a chance to finally tell a story that has been unfolding quietly for decades. This webinar focuses on the platform that has played a major role in the industrial biotech, but most people outside the field rarely have even heard about. So it's Trichoderma reesei. A lot of everyday products from laundry detergents, textiles, food flavors, colors, different ingredients, they all trace back to this microorganism. And at Onego, Trichoderma reesei sits at the core of our efforts to design the more resilient and sustainable food system. For precision fermentation, the real question now is scale. Is it competitive? Is it reliable? Does it perform at the standards of the global food manufacturers? And this is why we have created this series, The Power of Trichoderma reesei. At Onego, we want to talk openly about the technology itself, something that is still quite rare in food and biotech. Not because the fields would lack ambition, but because many platforms have not yet been validated at scale. So in this first session, you will hear two expert presentations. They're going to be 20 minutes each. First, from our dear guest, Dr. Sharief Barends, the head of R&D and fungal genetics at IFF. And after that, from my co-founder and Onego Bio CTO, Dr. Chris Landowski. You can, and we encourage you to submit questions at the Q&A section of the webinar. You can do that throughout the session, and the experts will answer them at the end of it. So with that, I will hand it over to you, Sharief, to get us started. Thank you.
Thank you, Maija. Good morning, good afternoon, good evening, everybody. It's a pleasure here to present IFF's powerhouse, Trichoderma. I hope you can see my screen. And then I'll just get started. Let me get my cursor, my pointer. So my name is Sharief Barends, as Maija already introduced me. I'm head of research in Leiden and the head of the global fungal genetics team that spans the entire globe from China all the way to Palo Alto, California. And I like to start always with this beautiful picture that I stole from a very nice YouTube movie. You can Google it. It's called Fungi, Web of Life. It's narrated by Björk, the famous singer, based on Merlin Sheldrake's book. It's very inspiring. Trichoderma and fungi are broadly used, and I just want to tell you a little bit more how we use it at IFF. And we started that journey decades ago. This is a little bit on IFF. IFF is a fairly large company. It stands for International Flavors and Fragrances. We have formed the current IFF organization through splits, merger, and acquisitions. It's currently a close to $12 billion sales company, and a lot of that money we reinvest in R&D. Sorry about that. There's a lot of products that we'll show here that we are touch upon people's lives on every day, and a couple of those are based on enzymes. For instance, the cold laundry products, 50% of those contain our product. 25% of the chickens around the globe are fed with IFF enzymes, and so on. So those enzymes, we develop them in the Health and Biosciences unit. IFF has 4 business units. It's Taste, Food Ingredients, Scent, and Health and Biosciences. And all the enzymes that we produce, engineer, is under Health and Biosciences. So we have a global enzyme innovation workflow, and that contains 3 major steps. It's discover, design, and develop. We have a discovery team in Shanghai, China, where they do enzyme prototyping on naturally existing enzymes. And they do that for all the different business applications for grain processing, home and personal care, food, biosciences, and animal nutrition and health. And what I always say is, Mother Nature created an enzyme, but not a dishwasher. So we need to tweak that. And that's what we do in the design phase, which is mostly coordinated and structured around Europe, with its center Leiden for the high throughput protein engineering. Here we design non-natural enzymes and to tweak it for the different customer applications. Then those lead candidates go to the US, where both East Coast and West Coast, we have teams that are excellent in engineering and operating cell factories, because at the end we need to produce those enzymes. And in all these steps, we use microorganisms to produce those enzymes. We have in our IFF portfolio, we have bacterial microorganisms, and we have a bunch of fungal microorganisms with our workhorse juggernaut Trichoderma. Trichoderma is an old one. Trichoderma was discovered in the Second World War on the Solomon Islands. It was taken off a canvas stand and was taken back to the US because people they found it interesting that a microorganism apparently secretes enzyme to digest canvas. Later on, it was published by Mendels and Rees as the Trichoderma reesei that they isolated, but not much later, they discovered that it was not Trichoderma reesei. It was a really separate species, and in honor of Elwin Rees, they called it Trichoderma reesei. So the mother of all Trichoderma is Cum6a. It's where it all started, and specifically in the 70s of the last century, there was an interest to further develop Trichoderma in a hyperproducer of cellulases. In the 70s, we had the oil crisis, and they were already looking for alternative ways of, for alternatives for fossil fuel, bioethanol. That was already happening in the 70s. And there are two major branches from QM6A that all have the same purpose to make Trichoderma more efficient to produce and secrete enzymes. And you see that, you know, in this classical strain improvement, that sometimes same mutations, they pop up independently, you know, and these are lead candidates for intensive research that happened over the years in academia, but also in industry. At IFF, we chose this RLP37 as a starting point for proprietary Trichoderma strain development for different purposes. In brown, we started first removing background by gene deletion. It was classical targeted to make the sugar that we feed the organism flow more to the product that we needed. Later on, we focused more on viscosity reduction, and I will go a little bit more into that. Leiden is the high throughput flagship for protein engineering. So we also developed Trichoderma in a way that it can handle high throughput workflows and configurations, microtiter plates, liquid handling, etc. And this is very different, but tied into the Trichoderma commercial strains. In parallel, we also moved Trichoderma into even higher productivity strains, classical, and even now we move to modern ways of strain engineering. And those learnings from the background removals, we now combine with these productivity improved strains into that new and latest lineage that we are exploring and using at IFF. So why Trichoderma? Trichoderma is safe, GRAS, EFSA approved. But what is very important, why Trichoderma is probably unrivaled when it comes to efficiency, is because it has uncoupled growth from production. It has an unparalleled induction system, and very importantly, it scales. So if we look at this graph over here, then in the typical fermentation, fat batch fermentation, we put in dry cell weight, and at a certain point at triggers, which is a oxygen decrease or CO2 decrease, we start to feed. And now you see the dry cell weight keeps constant while we start to produce enzyme. So that means that all the feed, the sugars that we are adding, does not go into biomass, but it goes into final product, which is very powerful. And that's why Trichoderma is probably unrivaled when it comes to carbon efficiency. Another example, what I said about unparalleled induction system, is that if you make a genetically modified fungal system, you need a strong promoter. And this is another fungus that we have in our toolbox. There you see that historically this, or naturally, this organism produces a little bit of a lot of different enzymes, while Trichoderma, wild type, makes a lot of a single product. In this case, it's the cellobiohydrolase 1, CBH1. So we can take this promoter, link it to a gene of interest, and then produce a lot of it. So, and you cannot make it work in different organisms, like to the level of Trichoderma. I want to go a little bit more into viscosity reduction. And why is viscosity reduction important? There are some key fermentation parameters that make the product, that make the cost price of the product and the best use of our equipment to the nameplate capacity that we want. That is yield. I touched upon that. So how much protein do I make? How much grams of proteins do I make per gram of carbon? But then there's another one, and that's volumetric productivity. And that means basically how well am I using my fermenters, the steel in the ground. And that's defined by how much protein do I make per hour per nominal volume, cubic meters. And that's defined by the number of cells, the dry cell weight per broth. And this is limited by oxygen transfer. And it's defined by the rate of the organism. So how fast is this organism making product per defined time? Now you can influence that by adding more cells into a tank and by not limiting the oxygen transfer, by playing with the viscosity of that organism. And we did so. So this is just a snapshot, well, it's a piece of art, if you ask me, of different morphological mutants of Trichoderma, proprietary strain, where we did all kind of different mutagenesis efforts, classical, but also targeted, where we measured altered osmotic sensitivity. We did sieving experiments, but also scoring microscopically to look for morphologically changed Trichoderma with a reduced viscosity while maintaining productivity and growth rate. So these are the do not harm factors that we don't want to touch. We don't want to lose productivity. We don't want to lose rate. But we want that Trichoderma filamentous fungus to reduce its viscosity by changing it morphologically. So we screened, and this is an effort we have done and we are doing. But back in the old days where this is 10 years ago or something, we screened 100,000 mutants. We had no techniques like Cas-CRISPR. We were not as high throughput as we are today. We did that through chemical mutagenesis, insertional mutagenesis, and this is the success rate of finding a reduced viscosity mutant. So chemical mutagenesis is very effective, 3 times more effective as insertional mutagenesis. However, insertional mutagenesis retains productivity better. Remember, we don't want to reduce productivity. We don't want to give into growth rate. So a combination of these kind of mutagenesis strategies gave us some mutants here, SFB3, SSB7, MPG1, SEB1, that show specifically here, even on plate, you see that from the parent, you have a reduced, restricted growth on plate. And you can even combine these mutations. Sometimes they combine, you have synergistic effect, sometimes they don't. It's very random and it's very difficult to predict upfront what's happening. So you just need to run these experiments. Now, do we run those experiments? We run those in our tanks, and we change the setup of the tank so that we can monitor for reduced viscosity, because the morphology on plate can be quite different than the morphology in a tank. So what we typically look at is a reduced requirement of steering. This is the RPMs that the impeller has to put in to keep up the carbon addition rate high and to keep the dissolved oxygen or low and keep the dissolved oxygen high. And you can see, for instance, for SLB3 or a combination here, that you reduce that requirement of the impeller steering speed. And again, what I said, like sometimes mutations combine, but you cannot predict it. It's like SFB7, when you look at it, it does not look much different from the parent. But if you combine it with an MPG1, and I don't show that MPG1 in isolation here, I don't have the data here, but you see that surprising synergy that the RPM does not go up a bit. So that's one of the factors that you have to run the experiment to do it. And sometimes we find surprising mutations that are under-characterized proteins, but sometimes it makes sense, like on the membrane part of Trichoderma. You can imagine that if you change the morphology of the membrane, you change also the viscosity of the cells. You make them less fragmented or more fragmented, less filamentous. But then also we find mutations that impact protein glycosylation. We did not expect that. So and again, the learning here is mutations combined sometimes, and we can go up to a much increased volumetric productivity, making much better use of the tanks. Another big effort that we apply at IFF is we always look for increased productivity of Trichoderma, so reducing the cost per product. And this is an example that this is an induction system of the promoters in Trichoderma. It's complex. It's amazing how nature invented it. I will not go into detail of here, but there are some actors here and repressors that in concert coordinate expression of the enzyme in a concerted way. So because you need the balance of those enzymes to degrade biomass in the natural habitat. So we use those promoter systems, we hook them up to the gene of interest, but we also try to understand and better use all these effectors along the way. So for instance, for the cellulase promoter, CBH1 promoter, if you reduce the carbon because it's carbon repressed and you add an inducer, the transcript level goes up a 10,000 fold. So that's the power of protein induction in Trichoderma. VTT discovered a very important factor, ACE3, that plays an important role in the cascade of inducing the CBH1 promoter in Trichoderma. It was a key regulator that was discovered in QM6A. Later on at IFF, we discovered that along the way, a tiny little C-terminal truncation happened along the way. So we teamed up again with our friends at VTT to better understand what that truncation did. So that's a truncation of about 10 to 17 amino acids at the C-terminus that happened in the lineage. And then in parallel, we discovered that the gene that was annotated was incorrectly annotated because it's quite complex in eukaryotes with introns and splicing of exons and everything. So we discovered that the gene is actually longer than what was described in literature. And that combination of the full length with the truncation here, we found that Trichoderma does not need the inducer anymore. So it can already, and I'll just focus on this L version, it can almost produce as much just on glucose. So it becomes inducer independent. But you see that adding the inducer still helps. So apparently now we have a double induction system, both on the glucose part and on the inducer part, which in this case is lactose, but there are other more known potent inducers like sucrose. So the N-terminus is required, a full length. The truncation is absolutely essential for the full induction and having that glucose effect. And then some introns are not required. This is the best version, and we connected that to an appropriate promoter. And what you see here is this is in our tanks at IFF, is that whereas you have production on glucose in the absence of this ACE3 configuration, you hardly have expression. Yeah, but if you add that ACE3 configuration, as I described in the previous slide, just on glucose, it is almost as good as on the potent inducer sucrose. But adding them both, the sucrose and the glucose with the ACE3, you get more. And you can see that here on the gel as well. In a normal situation where you don't engineer the strain with this ACE3 version, you hardly have expression. But then when you add that ACE3 version and the glucose sucrose, you really get the best benefit of it. So it increases productivity 23 to 60% with the inducer compared to the base strain where we were. So in a nutshell, Trichoderma at IFF, we want to go faster, better, more, and cheaper. So faster, technologies like Cas CRISPR, ONT, they are now integrated in our in our daily life. We use it in all the workflows, wherever it's protein engineering, discovery, or develop those traits into products. We want to make better products, improve product quality and stability by mitigating the background. And Chris will tell some more about it in his presentation. We want to go further, more. So every year we're expanding our enzyme portfolio, whereas traditionally Trichoderma was fermented at more acidic pHs. We now have the whole scope of pH, acidic to more basic and neutral, and that opens up options and possibilities to expand that enzyme portfolio. And we are, again, continuously improving to drive the cost down. So with that, I would like to thank you for your attention, and I will give the floor to Chris. And I'll stop presenting.
Okay, well, thank you, Sharief. That was really insightful, and yeah, very great to get the wonderful overview of what's going on at IFF. So that's okay. So thank you all. Just a little bit about me. I'm Chris Landowski, CTO co-founder. Of course, I also studied biochemistry and pharmaceutical science, and I got into working in industrial biotechnology when I moved to Finland. I worked at VTT Technical Research Center and was working a lot with filamentous fungi, specifically a lot of Trichoderma reesei. And also in that time, we started working, traditionally, as Sharief just said, there's been a lot of emphasis on the industrial enzymes, but we also we moved that into working with cellular agriculture and egg and milk proteins. And we started doing that in about 2016. And that gave me the opportunity in 2022 to spin off with Maya Onego to commercialize and work and focus on egg white proteins that we are working on, such as Bioalbumen product. But for me, it's an interesting path. Now I'm just showing in the background, you see a rendering of our future factory that will be in Jefferson, Wisconsin. So we have signed a purchase contract for the land in Jefferson, Wisconsin, and we have already completed the land surveys. This is a greenfield project, and we are now selecting the partners for the front-end loading 2 engineering work. And so interesting that the U.S. Department of Agriculture has also now given us a green light to support financing of the project. So of course, this is super exciting to be able to get into Wisconsin and to start in the future being able to produce our product there. And yeah, so as Sharief was just talking about precision fermentation, it's a little bit different. So enzyme industry is, of course, focused on enzymes, and there are, of course, mixtures of many different products in that mixture. However, precision fermentation is a little bit more refined in that we are actually now talking about ingredient proteins or fats, lipids, pigments, other things that are more precisely made as one ingredient from that. But we do kind of build upon all the decades of knowledge and expertise that the industrial enzyme industry has used. So we're just kind of moving into an extension of that. But there are some small differences that we have to pay attention to in precision fermentation. And one of those things, of course, since we're making a food ingredient product directly, we need to also pay a lot of attention to kind of the strains that we use. Are they food safe for EFSA, for the FDA? And also, are you able to work with them, and do they have good toolboxes available? So there's sort of a list of those kind of strains that would be appropriate. Of course, in the longer scheme of things, we do as many other industries would need to have a lot of output capacity of the strain, so they'd have to be able to robustly secrete a lot of protein. And then since we are also very conscious of costs and the purity of the product, we try to grow on very cheap, easily available raw materials such as glucose. And then also we also don't want to use more complex things like inducing sugars or anything like that. And of course, the system that you choose has to be ultimately very scalable, because for this industry, for food proteins, we're talking about thousands or tens of thousands of tons of product that are needed for that application. And of course, when you scale up, all those things have to be quite manageable so that the processes are easy to do and that are very simple upstream and downstream. So here are some very traditional protein production hosts. And of course, they're anything from bacteria to yeast to filamentous fungi. And really depends on the product that you're trying to produce, which system that you would look into. Certain enzymes might work better in different hosts here. And that, of course, is true. And traditionally, a lot of the most common things are the bacteria and the yeasts. And of course, they're really common and they're easy to access. So pretty much many entry-level companies can get access to them and start using them for their purposes. Many of the yeasts, let's say Pichia pastoris, for instance, is a pretty good secretor of protein. And so, they're also used in the medical industry to produce insulin or other biological compounds. However, there's an example that was published about phytase being produced up to 22 grams per liter. So from the literature, this is kind of the highest value that we're seeing. However, contrasting this to the filamentous fungi that Sharief had introduced, Trichoderma, for instance, is also a very high capacity secretor, so over 100 gram per liter that is sort of published. Of course, this is a very efficient host in that it's using a very low amount of biomass to do that. And also is not as filamentous as it sounds. There are also industrial strains that have now low viscosity, and Sharief had nicely shown that. And here's a kind of a case example of a bacterial phytase in the industry. So many of the major players in the enzyme industry, BASF, DuPont, Novozymes, Royal, have used filamentous fungi, Aspergillus and Trichoderma, for being able to produce a phytase. And it's obviously because, this is a very competitive industry. If there's 4 companies, 5 companies trying to produce the same product, so what is their advantage is probably that the cost effectiveness of the product is one major factor. And as an example of this is that the DuPont company early on has been working with a yeast here, and that was maybe their first production strain for the phytase. And early on, then they switched over maybe in 2012 or so to start working with Trichoderma to be able to produce the phytase in order to be more cost effective. So just industrially validating that fungal platforms are extremely good. Okay, and then a little bit about Trichoderma reesei and why it's very good at what it does. A little bit further view of what Sharief talked about, but more this is going to now dive into more like fungal biology and its history over time, evolving over the last 40 million years to secrete hydrolytic enzymes to break down the plant biomass, because it's living on dead plant biomass that's in the forest. And if it's living on some wood, the wood is cellulose, it can't access that sugar that's there. So its only hope is to secrete enzymes to break down the cellulose to create a sugar, a simple sugar that is absorbable and kind of usable by the fungus. So it has to secrete enzymes, and that's part of its lifestyle. Over 40 million years, it has figured out very well, how do I secrete as much enzyme as possible to get the sugar out fast, because I want to grow. And that's what it has done over the years. And a lot of these properties that were talked about make it very amenable for growing in a bioprocess and to scale up, because there's kind of relatively low amount of biomass that is not very demanding. It's possible to grow into very large scale cultivations, let's say up to 500,000 liters, although typically something in 150, 200,000 liter range. And of course, it's a nice system in that you, of course, are secreting the proteins into the culture media, and it's not like a bacteria that you have to break up the cells to do the downstream processing. So this is a much simpler system when you're thinking about the secretion end of things. All right, and just diving even further deep into the cell biology of this. So how is it so good at this? Well, it's been evolved for secretion over those millions of years, and the fungus, of course, is very adapt to sensing its environment. So if it's an environment where there's a lot of simple sugar around, that's like glucose, that's its preferred carbon source, preferred food. It will just absorb and take in all of the glucose in the environment, the simple sugar. So it just eat that and live off of that. But when that runs out, it needs to look for other things. So for instance, if it's sitting now on some wood, it'll start secreting enzymes. So it has to know what's in its environment, how does it adapt to that environment. So it will definitely then start secreting enzymes, cellulases, hemicellulases. In order to do that, it has to really highly ramp up its secretion pathway. So from the point of view of the cell, it's now going to be under more stress because secreting a lot of protein becomes stressful for the cell. But Trichoderma is extremely good at tolerating and adapting to high secretion load. So it has one of the best, most robust unfolded protein response, and that's something that how it adapts to secretion stress. And what it often does in this case is it massively upregulates proteins in the endoplasmic reticulum, the ER, that are able to fold proteins and help the proteins being made in the ER, folding them and sorting them in different places. So that can happen very well in Trichoderma, up to maybe 50-fold increase in those enzymes that are produced, foldases and other things. And also it does expand dynamically the ER space. That's actually where all the proteins are being folded and made. So it can enlarge the ER space also by 2 to 3-fold, because it's just starting to increase the volume. It's like making the road wider. It makes the road really wide, so just to put forward all the cars on the road so it can secrete more protein. And it also upregulates the vesicle trafficking mechanisms. So in Trichoderma being a more complex organism than yeast, so it has maybe 3 times as many genes in the genome compared to a yeast. So it has more tools in its toolbox to support high secretion. So there's maybe 3 times more secretion-related genes compared to Pichia. So it's very highly specialized for secreting. And as you can see here, the cell is polar, which means it has one end. And on the left side of this, there's a tip, the fungal tip and of the filament, and that is where it's secreting proteins from. So it's kind of directional. Things are going from right to left in this case. And those little vesicles are containing protein that it's secreting, let's say an enzyme, and it's moving through the Golgi. And it's kind of a special setup of the Golgi that is really put in stacks, so that's very efficient, and then secreted out the front tip. So this is kind of engineered, I guess, for high capacity output. And then one really other important feature to mention is that the optimal secretion occurs when it isn't very much growing. It's sort of static, sitting there, putting all of its energy into secreting enzymes. And this is one feature that we were able to take advantage of in our bioprocesses. All right. So now taking the Trichoderma system that is, of course, really wonderful from the enzyme industry, how do we now take this into food protein production? And there are some other kind of challenges that we had to address early on. So these are some technical developments, like how do we remove protease activity? How do we remove the backgrounds proteins from being secreted? We don't want to use inducer systems, so we want an inducer-free system, and we want very inexpensive carbon sources. Those were some of the technical challenges early on. So when I first came to VTT, I started working on a project with a pharmaceutical company, and they were making very sensitive biological compounds, growth factors and enzymes, and let's say monoclonal antibodies. And in this case, we worked a lot on trying to identify proteases. And proteases are enzymes that break down proteins. And that, of course, is not very much valued. It's a negative thing that fungi would normally do, but we have identified a lot of these proteases kind of genetically and then remove them from the genome. So in this case here in the article, I identified 7 proteases and genetically removed them to remove that protease activity. And we were able to reduce the casein proteolysis by 30-fold by addressing that. So in this sense, from this project and we were able to then come upon, even 15 of the most critical proteases and even remove them from different strains that we had. So in some sense, we have mastered protease reduction in Trichoderma, which also expands into any other product that we produce. That's also can be beneficial in many circumstances, depending on the product that we make. The other really important topic that we focused on was, as Sharief was talking about, the need and desire to produce on a glucose sugar source without inducers, because we want to have a very simple setup where we just feed glucose to the cultures and just get our specific protein being secreted and not having a lot of background. Whereas traditionally, if you have a very complex media, so here a spent grain lactose media, you will get an enzyme mixture. And the enzyme mixture obviously is full of a lot of different proteins. And we really would not prefer to have to purify that and purify our product from that. So we have here two different examples, a CALB lipase and a cellobiohydrolase, in which we were able to employ a new strategy to get pure protein production without much background. And that has big advantages. Of course, as Sharief had alluded to, that we can uncouple the growth from production and then channel all the sugar, all the carbon from the sugar into protein and not biomass. Because for us, kind of one of the most expensive things in the process is the sugar that is going in. So it can be a major portion of the cost. So how do we most efficiently use our sugar to make protein? And at least in some publication, you might even be able to produce at least 20, maybe 24% of the sugar can be converted into a protein in the article that was found here. So that is our goal, is to try to channel as much of the sugar into the product. And we can do that just by kind of growing up enough biomass that you need and then start feeding it at a limited rate so that it's just maintaining that same biomass. And then this fungus actually evolutionarily gets excited in promoting expression. So we can then use the natural promoters of Trichoderma to secrete our proteins of interest. So this is occurring when you don't grow and you're just secreting high amounts of protein when it's sort of static. And a lot of the other organisms, let's say yeasts and bacteria, are more prone to grow in a more linear fashion and grow a lot of biomass in the tank, which of course is not very efficient or effective when you're trying to be cost effective. Here's an example of Trichoderma reesei and Pichia pastoris, a yeast. And again, these are just the numbers coming from these publications in a way. It's a 100 gram per liter case. So these are like best case scenarios for both production systems. So 100 gram per liter production for the Trichoderma and 22 gram per liter production for the Pichia. And just assume these are whole broth titers. So whole broth considers the amount of biomass in the tank as well. And one really important point I want to make is that, go beyond titers and just think about what is the output of the system. I mean, how many tons of product can be made in the tank at the time? So for the Trichoderma case here, you're able to produce 10 tons of dry product, whereas if you had been using Pichia, you would have maybe 2.2 tons of dry product from the same enzyme you were going to make. Let's say this was a phytase enzyme that was mentioned earlier. This would be the case. So you produce way more from that same biomass. And also, I just comment quickly that, of course, when you have a Trichoderma cultivation, since you have a lot lower amount of biomass, the conditions there are not as demanding. There's so many fewer cells to feed. So the cooling power needed, the oxygen needed are all a bit less because it is not a very demanding process. Whereas with Pichia or some other yeast, you may accumulate a lot of biomass, which of course becomes very hot, and you have to cool that biomass, and you also have to make sure it gets enough oxygen. Or in some cases now, you also have to use methanol to induce the expression, and that of course is a flammable and toxic compound to be putting into the tank. So from this, just pointing out that always think about how many tons of product does your system make. And now translating that into more like, what is the cost impact of the selection of my platform system? So just taking the numbers from the previous slide in terms of 10 tons versus 2.2 tons. And then just for illustration, putting a hypothetical cost of batch. How much does the batch cost to make? So for instance, at 100,000 liters in that scale, maybe it's $50,000. And when you start then dividing dollars per kilo with Trichoderma at 100,000 liters, you would have about $5 per kilo, whereas the Pichia would have maybe $22.5 per kilo cost. And with the Trichoderma system, as I mentioned, it's a lot easier to scale that up into bigger volumes. Whereas here's an example of what it would cost for the 250,000 liter cultivation, and you could probably bring the cost down to around $4 per kilo as an illustration. And these larger scales are not that common for yeast and for Pichia. It's a little bit demanding. So of course, with a filamentous fungus, you can go larger volumes, and of course, see the benefits of the productivity and the volumes that you can produce. So you're getting to very low cost per kilo. And that's what you can do with the filamentous fungus. And that is why you see the enzyme industry using it, the precision fermentation industry using it. And that is why it is our preferred organism at Onego. Okay, well, thank you. And I hope to talk to you again in the future. And please send your questions along and we'll get along answering them. Thank you.
Okay. Yes, great. Thanks a lot, Sharief and Chris, for these great presentations. So we have got some questions here. Let me just pick some good ones for you. So, okay, well, we'll just go in some order here. If Trichoderma reesei is so efficient as you describe, why isn't everyone else using the same platform? Well, I think that's a good question. Who wants to start?
I can start and then Chris can add to it. I think they are. You know, like Chris showed in his table, there's a lot of companies using Trichoderma and they onboarded the organism long ago and then they deviated into their own proprietary strains for different use. We know our friends at competition are using it and we surely do ourselves.
Thanks. And Chris?
Yeah, I mean, I think a lot of people will like to use it, and it's just, of course, the proprietary strains that the industry have are very excellent, and there's just no way to access those things typically. So if you're a new company coming into the field, you're kind of harder to find. And of course, VTT has a good strains as well, so they're often a place to go for people who would like to work with Trichoderma. Of course, you can also look at a culture collection to get Trichoderma, but you know you're going to be decades behind trying to develop those strains. It'll take you a while, so it's good to get access to excellent proprietary strains in order to make this work.
Yep. Okay, thank you. What makes production of industrial enzymes and food proteins different, and how transferable is enzyme infrastructure to the food-grade proteins? If now Chris is starting. Okay.
Yeah, in this case, I think, as I mentioned earlier, everything is pretty much similar. I mean, we're using the same organism. You know, we have the same bioreactors, the same relatively same scale of working on cultivations. The only big difference may be that we are needing often to purify our product a lot more because it's going to be a food ingredient. Whereas in the enzyme industry, of course, there can be a lot of other things in there, and it's just fine because it's an enzyme. It is doing some enzymatic activity. Our products, from a food ingredient, having function, they are part of the bulk component of the product, and they have some other kind of functionalities that they foam or they gel or something like that. So we do just want to have our product in a pure sense produced.
Yeah.
Yeah, just adding to that, you know, it depends also on the application. And like what Chris is saying, sometimes you want like as pure as possible product. And sometimes it helps if you have a little bit of background activities. So at IFF, we have dedicated strains for exactly those purposes and applications.
Okay, and next one, what is the scale of your current production, both in liters and thousands of tons in your current and planned factories, both at IFF and Onego? So Sharief, if you start this time.
It's in the ballpark what Chris showed, and I cannot say much more about it.
Yeah,
Yep.
and then at least in our Onego factory, I mean, we're talking about like 150,000 liter tanks, and almost 2 million liters of capacity overall. That is what we're aiming at in the Wisconsin factory, and it's just a thing that for us the ingredient is that we need an egg white protein. And that requires thousands of tons of that product to be made. So we need to basically dedicate that whole factory capacity just to one ingredient. Maybe that's a 5,000 or 6,000 ton facility.
Yeah. Okay, this is a good one. So 10 years from now, what does success look like for Trichoderma reesei in the food industry? So is it going to be a niche solution, or do you see it becoming a core part of how food is produced globally? Chris, maybe you take this one first.
Yeah, I would just say that, of course, it really highly depends on the product you're making. Of course, I don't want to always think that Trichoderma is great, and I think a lot of the nice proteins that are really nicely folded and easy to produce, globular things, they produce really well, and that's true. So in terms of egg and milk proteins, I think it's an excellent choice for like whey proteins, and for egg protein like ovalbumin, excellent choice. They're relatively easy to produce. Whereas, if you have something like casein, where it requires lots of post-translational modifications, phosphorylation, other kinds of different activities on the protein, it may be challenging, and sometimes with filamentous fungi, or if you need a specific glycosylation pattern on your protein, it may be easier to work in some other organism. Or if you don't want glycosylation, you go to a bacteria. So like Sharief said, there is like a whole platforms out there that are kind of customized towards different products. And I would not like to generalize on anything that this can do it all. But if you're just looking at bulk output, production at a good cost, and the protein allows for it, Trichoderma absolutely is the king there.
Yep. Anything to add, Sharief?
Well, it's a choice, right? In 10 years from now, we don't know how the planet's going to look like. And if you want a sustainable bioeconomy solution, and ovalbumin is a very nice example, as a alternative to egg white, casein, alternative to milk, it's far out. But 50 years ago, we were dreaming of these kind of things that we're doing now. And if you are committed to a more sustainable planet, then this bioeconomy is probably the right choice. So we're only going to invest more in it.
Yep. Okay. Yeah. And okay, how does Trichoderma compare to other filamentous fungal hosts in producing industrial products in your experience? What do you think are the main attributes to the difference, in either biologically or evolutionary or industrial development wise?
It's a good one for Sharief.
That's a big question. So let me start that Trichoderma is not the only organism we have in our toolbox. We have many more, but by far Trichoderma is the most commonly used at IFF and probably also at other companies. There were a lot of questions in one. So it differs from other fungi in that Trichoderma is unique, that it has uncoupled growth from production. So unrivaled yield on glucose. There's no other organism that has that phenomenon as Trichoderma does. Trichoderma is unique in its natural habitat. Its strategy to eat dead biomass is to produce a lot of a single enzyme, whereas other fungi, they produce many different enzymes, but a little of it. So the genetics in Trichoderma is really very nice to have. So those are the big two attributes that makes Trichoderma very nice. And like what Chris said, the growth medium and production medium is almost synthetic. It's super simple.
Yeah.
Okay, here is a very technical question for Sharief. Are the mutants with lower viscosity due to gene inactivation? The knockout of the hit gene would give the same result, or point mutations that change the functionality of the gene product?
All of the above. Everything. Yeah, that's a very good question. So we find a mutant, we recreate the mutation, but also the alternatives like up, deleting, inactivating, but also that, and then sometimes we find gain of function mutations. Sometimes it doesn't really matter. It's random, but yeah, that's a very interesting question. The gain of function mutations is the one that we like because they're very hard to predict.
Yeah.
And that's, for instance, that example with ACE3, the truncation of 17 amino acids at the C-terminal end, it was a gain of function because now we didn't need the inducer, we just needed glucose. You could have never predicted this.
Okay, this is continuing very interesting. So in your opinion, in the next few years, what will be shaping the next strain development directions for Trichoderma? More protease deletions, secretory pathways, safe inducible systems, and how much do we actually know already? And what is the next step to be improving food systems using Trichoderma?
Oh.
Chris,
you want to take that one?
Yeah.
Amazing.
Yeah, I mean, what is the future in that? Of course, is we're always trying to push more out of it. I think that if you think that, I mean, what it's able to do in nature, actually, or the kind of total enzyme production it's able to do, if we can channel all that to one product, and that's the numbers are really high in that way. So that is something very unique. Like as Sharief said, Trichoderma is used to doing that, channeling everything to almost one enzyme. So we can take advantage of that to the Trichoderma and producing other products. But, we would like to have, hundreds and hundreds of grams per liter on very, very efficiently converting, 50% of the glucose into product or something like that. Very efficient and very high production levels. We'd like to have like 300 gram per liter on 50% conversion, something, these are kind of like sky high numbers in that way. So that's kind of where we'd want to aim as a research group or people to be able to do such things to make it super viable for producing just about anything so that it's just all cost effective all around. So I think just from the raw output, it has all the machinery for doing that. We just have to enhance it and use some of the modern tools that we do have now. As Sharief said earlier, a lot of things were done classically and very traditionally with mutagenesis, but now there's like CRISPR-Cas and there's high throughput screening and other things that really do help us find the needle in a haystack much faster. So I think that there's a lot of kind of a wind behind us too to try to make, a very lean organism that just does exactly what we wanted to do, which is make a lot of protein.
Yep. Okay, let's speed up because I have a couple of very interesting questions still in here. First for Chris, is the continuous fermentation going to be a game changer for Trichoderma fermentation?
Well, I don't think that is so it is sort of already in a way that is sort of what we're doing, right? So like we are not draining it off, but as I say, we're keeping the fungus in a kind of static state that is just taking the carbon, sugar, and converting it to protein. Of course, you could drain off that and kind of they do this in enzyme industry already. I think they're called like draw and fill kind of techniques already, where you can still draw off the liquid and add more in. So that has been done in enzyme industry, and that can be done. That's just another tool in the toolbox if you want to go that route, but it requires a little bit different factory setup, I suppose. But I don't know, what does Sharief think about continuous fermentation? That would be very interesting to hear your perspective.
Well, it makes sense if you make one product, but if you have a portfolio of 150 different enzyme activities, you want to scheduling, right? You need to clean out the tank, come up with the next product. So, but it does make sense when it makes sense.
Yeah, because I think the idea is that you would have to dedicate that tank for a month just making one product, and is said if you have one product, it may make sense to do that. And we have already done some of these work in VTT. I think it was even 20 years ago already. We were looking at very long cultivation, so we know it can be done. We just haven't necessarily needed to go to that route at the moment, but it's a tool that we could partake in later.
Yep. Okay, we have a ton of questions flowing in, and I mean, I can already promise that we will answer all the questions later on. We will put all the answers to the websites. So yeah, I just want to end up with something that is a really beautiful one. So why did you personally choose to focus on your own career working with Trichoderma, and how did it happen? So just short answers, but I think this is a very nice way of ending this webinar.
Yeah.
Who wants to start?
Go ahead, Sharief.
I was born in Delft, where there was a yeast factory, and I managed myself to get an internship. And then, life happened. And then after that, after 15 years, I joined that company again because I like to do what we were doing. That was biotech. Before that, I never touched a fungus, and that's the first time in Palo Alto, California, I touched Trichoderma. And since then, I love to work with Trichoderma. It's a beautiful organism, and I never went away from it.
Yep.
Yeah. Yeah, from my side, of course, I'm from the United States, and I happened to marry a Finnish woman, and I came to Finland and started working at VTT. So then, of course, if you're at VTT in biotech, you're going to be introduced to Trichoderma because that is like the flagship research organism that we had worked with. So it just is a coincidence that I met up with a Finnish woman who's my wife, and I started working at VTT. Of course, before that, I was working on mammalian cell cultures and cancer biology, and I started working with filamentous fungi and found it extremely interesting because from a research point of view, there were so many unanswered questions. I mean, it's not as research topic as in mammalian biology, right? So I always kind of fell in love with the idea that I have a lot of things to discover. And as you can see, some of the publications that I showed are all major kind of findings in the field that how we could improve and study and understand Trichoderma just a little bit. But there's so much more there. And there's, as Sharief said, you can never anticipate or predict what might happen. So you do have to experiment. And it's a really nice organism to work with for many reasons.
Yeah. Yeah, I still remember when somebody was asking that, are there multiple Chris Landowskis out there? Because you actually have published so much, so it is interesting. But hey, time is running, so that was it. We need to end this for today. Thanks for everyone for joining. It was super fruitful. Special thanks, of course, to Dr. Sharief Barends from IFF for sharing your expertise here, alongside with our very own Dr. Chris Landowski. So this was now the first session of the Power of Trichoderma series. In the next one that is going to be in January, we will be exploring the manufacturing and scale up. So what takes to bring Trichoderma to industrial scale? We will send an invitation for the January session soon. So please sign up and also share the invitation with your colleagues and friends. We'll also publish a white paper that expands on today's topics. So stay tuned. We will see you in the next session. And meanwhile, happy holidays, everyone. Thank you.
Thanks all.
Bye.
Bye-bye. Thank you.